disease, or BRD, is the most common and costly disease affecting the North American beef cattle industry. In the broadest sense, it refers to any disease of the upper or lower respiratory tracts.
I have BRD. My daddy had BRD. My granddaddy before him had BRD. We all had BRD.
Among the many factors that can cause BRD, 70% are influenced by the environment. Stress-induced environments, including transportation, like transporting your cows from Texas to Montana, along with close quarter housing and wet or muddy pens, increase the probability of BRD. In this example, there are cattle that are kept in close quarters and in muddy pens, so the risk is higher for them contracting BRD.
Proper solution to reduce stress within the environment would be to locate cattle and properly manage pens. As an example here, we have cattle on grassland or pasture ground where it's more open, more spacious for cattle to run around, rather than being compacted together. Moving cattle slowly but effectively can also reduce the weight of environmental stress allocated with BRD.
In this picture from the St. Joe stockyards, you can see there's some pens that are overcrowded, and those are more likely to spread BRD than these that are less populated.
Within the cell lies a set of chromosomes specific to each organism. In each homologous pair, one chromosome is donated from the sire and one from the dam. Located on each chromosome are inherited genes that, in combination with environmental factors, determine specific traits in the animal. These differences in genetic code and environmental factors create variability within a group of animals. This plays a role in an individual's ability to fight off disease. Researchers have found specific genes associated with BRD to create selection panels to help genetically advance herds towards BRD resistance.
This is an example about how we can cure BRD in our herd through selective breeding. In this case, the brown M&Ms refer to the dominant or resistant trait. Green is the recessive trait, which is susceptible to BRD. So in our parent generation, we have a set of genes. Gene number 3 is the BRD gene. The mothers in our herd are homozygous recessive. We purchase a bull that is homozygous dominant. When we breed them, our F1 generation is 100% heterozygous. When our F1 generation matures to breeding age, we begin to breed them with another bull, which is homozygous dominant. This creates an F2 generation, which are 50% homozygous dominant and the other 50% are heterozygous. At this point, we can pull hair samples from our F2 generation and place them on a hair card. From there, we send them off to be genotyped at a lab.
Here we have our gene seed card that will contain our hair sample. We pluck the hair from a cow's tail and we put it on the card. Then we seal it and send it off to the labs.
They provide us with which animals are homozygous and which ones are not. So from there, we can use this information to be able to cull the herd and take out all the animals which are not homozygous dominant. From this point, as long as we continue to breed with bulls that are homozygous dominant, we should never have the BRD susceptibility gene back in our herd.